So, good afternoon, all the participants, faculty members from ISFCP, and our today's speaker, Professor Dr. Pravin K. Shende. So, welcome you, sir, on the ISFCP platform for the session 2, day 1. Sir, firstly, I would like to introduce our today's distinguished speaker. So, Dr. Pravin Shinde, sir, is currently working as a professor in the Department of Pharmaceutical Technology, formulation at NIPER, SAS Nagar. Sir has previously worked as NMIMS Mumbai for 7 years. Sir has hold the bachelor degree in 2001, master degree in 2005, and PhD in pharmaceutical sciences in 2009. Sir has 19-year teaching and research experience with 3 postdoc doctorate from University of Torino, Italy. Sir has worked in the number of research areas, including biosensors, nanosponges, nano bubbles, nanoflowers, and microneedles, uh, resealed erythrocytes, biocarrier drug delivery, DOE-based formulation, liposome, dendrimer, solid lipid nanocarriers, polymeric nanoparticles, and targeted drug delivery system, inclusion complexation, pre-formulation, and studies. Sir has 2 to 5 papers in different journal of international repute, 1 book published, and 10 book chapters. Sir also completed number of projects like SPER, YCMR, AICT, and government of Maharashtra project. Sir also has transfer of technology to his credit. Sir has 5 granted Indian patents, 1 international patent to his credit. He has guided more than 60 M.Pharm students, 5 PhD research scholar, and uh, and nominated in 2% scientist by Stanford University. So, sir, welcome you at the platform of ISFCP. Over to Sanjay, sir.
Thank you, San sir, for this introduction, and thank you, Dr. G.D. Gupta sir, for inviting me for this FDP. For today's topic, I just wanted to present the potential of nanotechnology in drug delivery system for multiple elements. So in this, mostly I am covering the different parts of nanotechnology formulations used in the drug delivery. Can you go to the next slide, please?
Okay, sir.
Okay. Then in this case, when we classify the nanotechnology in pharma, we always divide it into 2 parts. One is nano devices and second is nanomaterials. Nano devices mostly we use in the medical devices system, whereas the nanomaterials we are used for the delivery or dosage form. So nanomaterials we are classified into 2 forms. One nanostructured material and second nanocrystalline material. So mostly these materials are also somewhere interlinked, but nanostructure materials we mostly concentrating because these are used for viability, solubility enhancement, all this purpose. So nanostructure materials we are considering polymer based, non-polymeric and lipid based. In polymer based we have the dendrimer, then nanoparticles, micelles, drug conjugates, nanogels, protein nanoparticles. In non-polymeric, where we are having the carbon nanotubes, then nanodiamonds,
हाँ?
metallic
सर को मेल
nanoparticles,
की है.
quantum dot, silica-based nanoparticles, and lipid-based, we are having the liposome, then lipid-based nanoparticles in microneedles, and SLN, that is solid lipid nanoparticle. Now, if you know that there are more specialized systems also available in the market for the nanoparticles, like nano popcorn, nano flowers, then nano vapors. These are new types of the delivery system. So when we talk about the drug and delivery systems, okay, we have the API and so many things we are doing. But this is today's new version where the dye we are using as a drug. So in this case, I just wanted to give the information that curcumin in turmeric or methylene blue, this methylene blue was widely used in the SARS-CoV-2 inactivation. So in this case, the methylene blue directly act on the singlet oxygen, and then in presence of light, it completely inactivate the SARS-CoV-2 in 5 to 15 minutes. And if I want to ask about, means tell about the thing about methylene blue, the methylene blue is the dye which is commercially available in the market, and this is used for the methemoglobinemia treatment, where the brown color blood is converted into the red color blood by reducing the methemoglobin reductase enzyme. So, looking at this concept as a dye, we move to the phthalocyanine. This is the dye most widely used for the different treatments of cancer, then for the antioxidant purpose. So, we consider that in the literature somewhat mentioned this is because of the presence of the groups, it is widely used in the diabetes also. So, looking for that type of the literature, then we decided, okay, we will go with the phthalocyanine. And there we found that the phthalocyanine is the versatile dye. This is the structure similar to the porphyrin. This is micromolecule, microcyclic organic compound. Then it is having the very good antioxidant activity, and this is widely used in the treatment of Alzheimer's and cancer. But this is only the research proposed, there is no marketed formulation. So looking at all this scenario, we selected the diabetes mellitus, and in this diabetes mellitus, we consider that hyperglycemia, glucose fluctuation, and insulin resistance. And all this, if you see the mechanism, if there is an endothelial dysfunction, then diabetes vascular complication can also happen. So looking at this scenario, we wanted to establish the proper dose of the phthalocyanine, as well as we are looking for in the coming days the mode of action of the phthalocyanine. So, as I mentioned previously, there is no dose mentioned anywhere in the literature. So, we wanted to establish the phthalocyanine dose. So, this phthalocyanine we injected into the streptozotocin induced diabetic rat, and then we divided into the four groups animal, so negative control group, positive, and here we wanted to select two doses, 10 microgram per kg and 20 microgram per kg, which one is better for the anti-diabetic action. So accordingly, we found that 10 microgram per kg is highly active as compared to 20 microgram per kg. So in this, as I mentioned, here this phthalocyanine reduce the serum glucose level significantly. They reduce the ALP value also, and the reduction of the TPC and 1, and we found that in the histopathology there are regeneration of pancreatic beta cells, and from that we can say that the phthalocyanine having the very good anti-diabetic activity. So in this case, when we consider the phthalocyanine again having the solubility issue and so many, to convert that into the better formulation, then we wanted to use some nanotechnology based concept. So we selected three concepts. One is cyclodextrin based drug delivery system of phthalocyanine, then lipid based, and third one is the PLGA based metal nanoparticle. So in the coming slide, I am first describing this how we prepare phthalocyanine based cyclodextrin drug delivery system. In this case, we use the polymer condensation method. Cyclodextrin first form the inclusion complex with the phthalocyanine, and then we complex that with the hexamethylene diisocyanate around 70 to 80 degrees Celsius, and then there was a complex formation. After purification of the complex, that is in the form of the nanosponges, then we injected that into the streptozotocin induced rat, and same way we observe there is a very good anti-diabetic activity because of the reduction in triglyceride and cholesterol layer. Then if you see that in the diagram, there is a much more reduction in the blood glucose as compared to the individual drug, and again the regeneration of the pancreatic beta cell. Now, in this case, the purpose of the using cyclodextrin was to enhance the solubility, but again, there was the issue that the phthalocyanine coming out from the cyclodextrin cavity. So we wanted to complex it with the hexamethylene diisocyanate. That was the cross-linker we used. Then, as I mentioned previously, the second system was the lipid-based phthalocyanine system, where we use the different types of the lipids, and from that we selected the one lipid at the initial level and then make the complex with the cholesterol, because the cholesterol increases the rigidity of the liposome. So then we added the dye first, and this method we followed using the rotary evaporator, that is nothing but the thin film hydration method. So we got first liposome with this method, and then we added the calcium chloride solution, and then what happened here, after the addition of the calcium chloride, the structure changed completely, and this structure is in the form from spherical structure it is forming the cochleate, and this cochleate is nothing but a cigar like structure. So in diagram you can see the cigar like structure. Then the problem was that this cochleate doesn't have the NH group outside. So because of that, to add the amino group, we added this EDAC or EDC and NHS, and after treating that, we found that the NH group decorated on the surface of the cochleate. Then we add the concavline, and because of that, there is a conjugation of concavline on the surface of cochleate. And then here in the diagram, you can see that concavline A conjugated phthalocyanine cochleate. These formulations we injected into the rat, and then we found that there is a significant decrease in the blood glucose level, and there is a decrease in the oxidative damage, and because of here we can say that we got a very good anti-diabetic activity. Then third system, as I mentioned, we got the PLGA based metal nanoparticles. What happened in this case, when we look for the phthalocyanine having the very good activity, but there are some metals which are potentiating the activity of the phthalocyanine, and because of that we added the zinc. So in this case what we did, the organic solvent contained the PLGA and the aqueous phase contained the PVA, chitosan and phthalocyanine. All we mix together using the high speed homogenizer, and then we got the drug loaded nanoparticles. We then optimized it in the three square design, then characterize these particles, and after oral formulation we found that this also having the very good anti-diabetic activity. So these formulations we prepared, and here these are some characteristic parameter of this formulation. So in this case, if you see that the particle size of polymeric nanoparticle is less as compared to the metallic and lipid nanoparticles. Zeta potential of lipid nanoparticle is substantially less as compared to the other two. Entrapment efficiency. Now in this case, if I can say that the lipid having the maximum entrapment as compared to the metallic, because addition of the cholesterol improve the rigidity of the membrane. The drug release, I can say here, the polymeric nanoparticles form the matrix system, and because of that there is a longer duration of the release. Then here the release again that with the metallic nanoparticle, the conjugation is too high, and because of metal and this, there is a slow release, and the release all happened for the 72 hours. Then BGL level also, it is less in the metallic nanoparticles. Triglyceride also less in the metallic, and cholesterol, that is also less in the metallic. So here I can say that okay, there is a problem with the release and the entrapment uh, efficiency, but the zinc and phthalocyanine complex form the very good chemistry, and because of that we are getting the synergistic activity. So next, that as I mentioned, I am looking for the different ailments. So that was for the diabetes. Now coming to the withdrawal symptoms. The people mostly having the cigarette smoking behavior, and for the cigarette cessation, we require the mostly nicotine. And if you see that there are so many nicotine formulations are available, starting from tablet, chewing gum, transdermal patch. Okay, there is a need of new patch, that is the microneedle. So we work on that, and first time we use the combination of the drug, because this nicotine and variclinil in presence of each other shows the higher withdrawal symptoms, and sometimes there is a dependence because of the nicotine alone. So this combination therapy showing the better result. So in this case, how we prepare the nicotinic polymeric nanoparticles, where we use the aqueous solution as well as the organic. Aqueous containing the PVA and the organic contain the PLGA, and then start the stirring centrifugation, and then this polymeric solution then we pour into the female mode, and from that we receive the microneedle. So as I mentioned that here the polymeric microneedle loaded, here this is the nicotine and variclinil. So because of that, nicotine nanoparticles are present inside, and variclinil is present outside the layer of that microneedle array. So in this case, you can see that when there was the application of the microneedles, okay, so there are reduction in the withdrawal symptoms like tremor, shake, jumping, backing head nodding, shaving, scratching, grooming, all these. And here we can say that what happened? Nicotine binds to the nicotinic acetylcholine receptor, and then there is a dopamine release. But varenicline competitively binds to the nicotine acetylcholine receptors and prevent the dopamine release. And thus there is a reduction in nicotine withdrawal symptoms. So this combination, I can say that this is the one of the alternative in the coming days for the conventional therapy. This is quite interesting. I can say that as your topic of FDP is related to AI, so this is the one system we last year prepared in the lab, that is molecularly imprinted polymers for the detection of glucose in saliva. Here, this systems we prepared in the three form. One is cotton swab, second one is the paper strip, and third one is the polymeric film. So mechanism was like that: glucose interacting with the oxygen, and there is a formation of gluconic acid and H2O2. TMB is the dye. In presence of H2O2, it forms the blue radical cation, and this then detected by the RGB detector, and then it will show the concentration of the glucose level on the screen with the programming. So as I mentioned, three systems we wanted to prepare. So first one was the HPMC film. So we added a lot of that material with the HPMC and prepare the film. Then in the cotton sap, we use the MIP, that is the molecularly imprinted polymers, where first we are adding the template and then removing the templates, and these are behaving like the artificial cells, and they are having the more specificity toward the particular type of the active. So that active we consider here as the glucose. And the third one is the paper strip, that here also we use the MIP. This MIP again we prepared using the glucose. So first of all, we prepared the 3D printed case. So here I can say that we prepared the STL form of the first that design, and on that basis then we prepare the three this type of the box, and this was prepared using PVA, and then you can see that these are the dimension as per our requirement. So then we place the different types of the circuit, where we can say the first one was the LCD screen. Then second was the RGB detector, that is for the color detection. And this is the main, that is the Arduino Uno microprocessor. This widely used for the any type of the detection system. So I can show you this video also. There was a append of, in the append of this type of that cotton sap, you have to just place with the sample, saliva sample, and there is a change in the color, and this signifies that there is a programming, and this programming just calculate the concentration of the glucose in the deciliter, mg per deciliter. So accordingly, we validated the different types of this sensing system. In this case, we have gone through the LOD, LOQ, sensitivity, then accuracy, specificity also. I showed only small parameters. And three systems we validated, and these systems also showed the very good uh, validation, and there was not that much that we can say the differentiation. We didn't use any type of the standard, because the AccuTest and all these, they are using the blood, so it's not correlating that the saliva and the blood and that's why we avoided that one. So thank you. Any question?
Yes, sir. It was a great work indeed. Uh, this was product based discoveries.
Thank you. Here, I just wanted to tell you the
Yes,
AI
sir.
principle we can use here,
Yes, sir.
because the patient to patient variation or patient to patient data that every day or means every time they wanted to suppose record. So based on that, the system will collect the data, and the system will in the coming days show the data in the graphical manner, so up and down and everything, the accuracy of the result, everything you can observe on the screen.
Yes, sir. So this was actually validated therapy for detection of glucose. You have worked a lot, sir, in this thing. So this was great work. I would like to summarize. Firstly, sir has detected some. Sir has described some utility of nanotechnology in pharma sector, and majorly cover the therapies of diabetes using nano therapy. Sir has discussed the preparation of hallucinogens using different method, including cyclodextrin, lipid-based drug delivery, and PLGA-based nano-composite system, and describe their relative importance and relative utility in animal model, also in for regulating the glucose. Finally, sir has discussed the preparation of microneedle using nicotinic polymeric nanoparticles, and discussed their compatibility. Last one was very interesting. This was a discovery of indeed a biosensor based detection of saliva, where sir used swab based system, paper based strip also discussed, and polymeric fibers also discussed. Sir, this was indeed good work in the field of diabetes. In fact, our FDP participants will also get some great knowledge from these discoveries. This is actual discoveries, sir. You have discussed.
Thank you.
So, firstly, I would like to call director, sir. If sir is free, he will join. So I think sir is in an important meeting. So thank you, sir, for being with us on ISFCP platform for FDP. Uh, Thank you very much, sir. Good day, sir.
Thank you very much.
Good day.
Okay.
Good day.